In the late 19th century, German biologist Walter Fleming observed thread-like material within the nucleus of the cell. This thread could be stained with certain dyes and could be observed under a microscope. He also observed that during the cellular division, this thread splits and its parts move to the two daughter cells that are produced during the cell division. He called these threads chromosomes, which literally means colored bodies. Chromosomes are basically packets of genetic material, which is the DNA. The DNA is packed into these bodies and moved into the daughter cells during the cell division. And the best time to visualize chromosomes is during a phase of cell division called metaphase, during which the packing of genetic material is at its most compact stage. Our cells have a definite number of chromosomes, and each chromosome has a pair. Humans have 23 pairs of chromosomes, which means a total of 46 chromosomes. Out of these, 22 pairs consist of chromosomes which are similar to each other in terms of the kind of genetic material they pack. Thus, each one of these pairs consists of two similar chromosomes, which are called homologous chromosomes. The last pair, which is called sex chromosomes, is not identical in case of males. In males, the last pair consists of one X chromosome and one Y chromosome, while in case of females, the last pair consists of two similar X chromosomes. When stained with special dyes such as giemsa, the chromosomes show alternate bands of light and dark staining. These patterns of bands help scientists recognize the homologous pairs under the microscope. Micrographs, which means photographs taken under microscopes, are then clicked and pairs are shown side by side. This pictorial representation of chromosomal pattern is known as karyotyping. Each karyotype is unique to the species. Each chromosome has a special structure called centromere, which divides the chromosomes into two arms. The centromere may not be present at the center of the chromosome, which means one arm of the chromosome is longer and the other one shorter. The longer arm is called Q arm and the shorter one is called P arm. At the end of each arm, there is a structure called a telomere. Both centromere and telomere are essential for the structural integrity of the chromosome. During cell division, the genetic material duplicates, which means that each chromosome duplicates into two, and these two are called sister chromatids. Sister chromatids remain attached at the centromere till they separate and travel towards the two new cells that are formed at the end of the cell division. Thus, when we visualize the chromosomes during metaphase, those sister chromatids are seen attached at the centromere, and thus there are two long arms and two short arms that are visible in each chromosome. Now, with this much basic understanding, we can study the aberrations or abnormalities that can occur in the chromosomes. These aberrations may express themselves into genetic diseases, or they may be silent. The persons in whom these aberrations are silent are called carriers. Carriers may give rise to abnormal gametes, that is, abnormal sperms or eggs, and may transmit these aberrations to their progeny. Each species has a definite number of chromosomes. The chromosomes also exist in two sets, male and female gamete contributing one set each. Chromosomal aberrations can be of two types: numerical, where an abnormal number of chromosomes is present, or structural, where the number of chromosomes is normal, but there are some structural abnormalities in the chromosomes. Cells of each species have a definite number of chromosomes. The chromosomes also exist in two sets, mother and father contributing one set each. For each chromosome from one set, there is an homologous chromosome that comes from the other set. When there are more than two sets present, the condition is known as polyploidy. Polyploidy is seen in plants. In human beings, polyploidy is lethal, and such pregnancies result in abortion. When the total number of sets is normal, but there is an abnormal number of chromosomes within any of the two sets, the condition is called aneuploidy. We know that there are 23 pairs of chromosomes in humans. It is possible that there is an additional third chromosome present in any of these pairs. Then this condition is called trisomy. Most common trisomy is seen in the 21st pair, which results in the Downs syndrome. Other examples of trisomies are trisomy 18, which means that there is an additional 18th chromosome. Trisomy 18 results in Edwards syndrome. Trisomy 13 results in Patau syndrome. Another type of aneuploidy is when there is only a single chromosome in any of the pairs and its companion is missing. This is called monosomy. Example is Turner syndrome, where in sex chromosomes, which is the 23rd pair, only one X chromosome is present. Each chromosome contains DNA in a particular sequence. Structural abnormality results when this sequence of DNA is altered. In cases where any part of a chromosome is deleted, the condition is known as deletion. An example of such aberration is Cri-du-Chat syndrome, where a part of the 5th chromosome is deleted. In case if there is addition of genetic material in a chromosome, such a condition is known as insertion. Insertions are rarer than other structural aberrations. There can be a case where a part of a chromosome detaches and rotates by 180 degrees and reunites. In such a case, the ends of such a part will lie in the reverse direction compared to the normal genetic sequence. Such a condition is known as inversion. There can be cases where there is an exchange of parts of a chromosome with another chromosome, and it is known as translocation. An example of this is seen when such parts of genetic material are exchanged between 9th and 22nd chromosome. The chromosome that forms as a result of this translocation is known as Philadelphia chromosome, and it has been documented to be responsible for causing chronic myeloid leukemia, which is a form of a blood cancer. Another example of a different kind of translocation is when short arms of two chromosomes are lost and their long arms connect together. Such a translocation is known as Robertsonian translocation. In 5% of cases of Down's syndrome, a Robertsonian translocation is observed between one chromosome between 15th to 17th and the 21st chromosome. Primary causes of chromosomal aberrations are errors occurring during cell division like non-disjunction, advanced maternal age, damage to DNA due to various causes such as ionizing radiations, various chemicals or viruses.